Our next speaker, Terrence Sejnowski, is the director of the Computational Neurobiological Laboratory at the Salk Institute. He is a colleague of Roger Bingham. He's also one of the co-founders of, along with Roger, the Science Network. We're trying to get this Science Network off the ground, which would present, 24/7 science, be sort of a C-SPAN of science. And Terry is one of the people involved in that and has really been instrumental in advising us and all that. His book, along co-authored with Steve Quartz, who was going to speak today but can't for medical reasons. Terry will be speaking a few minutes longer on the topic of neuroeconomics in addition to what else he was speaking, because he co-authored that book with Steve, Liars, Lovers, and Heroes, What the New Brain Science Reveals About How We Become Who We Are. And Terry, your book came out actually when I was just finishing up The Science of Good and Evil, and there was a lot of really good material in there for me about the evolution of cooperation and what's going on in the brain, that kind of stuff. It's really quite a good book. We have it here. Anyway, so he got his PhD in physics from Princeton University, and he earned the Presidential Young Investigator Award from 1984 to 1989, the Fairchild Distinguished Scholar Award from 1992 to 1993, the Wright Prize in 96, the Hebb Prize in 99, the Neural Network Pioneer Award in 2002, and last year the Johns Hopkins Society of Scholars Award. He's a first-class, world-class scientist who also can write really well, which is always a nice combination. Please help me welcome Terrence Sejnowski.
So I have to confess something. The morning session was fabulous. I really learned a lot. In particular, consciousness is something that we're all familiar with. But in my view, consciousness is just the tip of an iceberg. And that all the heavy lifting that's done in your brain, allowing you to see, to make decisions, and move around, is all done beneath the surface of the water in your non-conscious processing. Now, Francis Crick and Christoph Koch created an acronym, NCC, for the neural correlates of consciousness. And I'm going to create one here today, because what I'm searching for is the neural correlates of non-consciousness, NC squared. And a good place to start is while you were sleeping, because clearly the brain's in a different state, an altered state. But it's not silent. There's a lot going on in your brain. In fact, electrical activity changes character, but it doesn't disappear. I'll be telling you more about that. But before we talk about sleep, I want to give you a feeling for modern views of how the brain is making decisions beneath your level of consciousness, to give you a sense for some of the new discoveries that have been made. And I want to tell you a little story about Sidney Brenner, who's a colleague of mine at the Salk Institute and was asked to give a distinguished lecture recently at UCSD. And he was saying that biology is very different than it was when he was first getting started. He said, now, the genes have created a whole new field called genomics. It's been very successful. But now the protein people who have omic envy have relabeled themselves proteomics. And students now are going around. When I was in school, we used to go to the library and read journals and books, and that's where the knowledge was. But now, of course, they sit at their computer terminal and they are able to conjure up all of these articles in their computer, and they call it mining the bibliome, or bibliomics. Now, what Sidney pointed out is that the biotech companies, of course, don't want to get left out of the act, and so, of course, they've created their own version of omics. They call it economics. And in fact, that's what's happened in neurobiology, that there has been in the last few years an intersection between the traditional study of how human beings interact in cultural groups and how decisions are made through economic decisions and negotiations and the machinery inside the brain. So we're going to take a little journey. And the experiments that these new results are based on, I'm going to illustrate with this picture that was published in Science magazine about 5 or 6 years ago. Just to give you a sense for how the different parts of the visual system are organized, and also the way that people think about the signals at different parts of the visual system. So this is the monkey, and in these experiments, the monkey is sitting in a chair. The monkey is reacting to a stimulus, a visual stimulus that will come in and activate a pattern of activity on the retina. And the monkey has been trained over many weeks and months of training in order to respond, for example, to a particular type of stimulus, say a picture of a frog, by pressing a button, right? You can't tell the monkey which button to press, but you can reward it if it presses the right button, or press a different button if it's a different stimulus. So here's the stimulus. It takes about 25 milliseconds for that information to be processed within the retina. Then it goes up to a relay nucleus, and there's another 5-10 milliseconds up there. And then it goes to the primary visual cortex. And we know from the work of Hubel and Wiesel in experiments just like this that the cells there respond very vigorously to specific patterns of light, oriented bars and edges at different locations of the visual field and with different orientations. But that's just the beginning. There's a whole sequence of events, the V1 projection to V2 and to V4, down the so-called ventral stream, and eventually to the inferotemporal cortex, where Christoph Koch presented you with evidence in humans that there are neurons that are so specific that they only respond to Bill Clinton. But that's now the end of the pathway for vision. There are projections from the back of the brain to the front of the brain in the prefrontal cortex. Now that takes about 100 milliseconds. But now the information about the stimulus is there, and the animal has to match it with memory and now has to make a decision about which finger to press the button. And that sets a whole string of action into play through the motor cortex and finally down to the fingers where the actual button is pressed. So what's wrong with this picture? You can respond in a few hundred, 200 to 300 milliseconds to very complex images. It's amazing how fast we respond. Well, what's wrong with this picture is, first of all, it's one way. You'll notice the information goes in and goes around this loop as if it were just following a sequence of events. But that's not how the anatomy looks. Every single one of these cortical pathways has a feedback connection carrying information backwards, which is being ignored here. But there's something else that's even more profoundly wrong with this picture, which is that normally when we go through the world, it's very rare when we're confronted with a sudden flash of light and are asked to respond instantaneously. It may happen if you're driving and there's something that suddenly occurs in front of you and you need to avoid it, but most of the time you're planning ahead. Things are happening through your own self-motivated behavior. You make decisions to go somewhere. You make decisions what to do when you get there. You're planning ahead. And that's reflected in these recordings from these different locations of the brain by what's called by neurophysiologists, but is rarely emphasized, it's called spontaneous activity. Activity that cannot be accounted for by any external stimulus. It's activity that's being generated by the brain itself. It's very difficult to study, however, because of the fact that we don't have any control over it. So most of what we know comes from experiments like this, where we create an automaton. We put the monkey into, and humans, by the way, are also subjects for experiments like this, but we force them into a mold to be a reaction system, to react to as a reflex to a particular stimulus. But that's not the way the brain was designed to work. We heard this morning that babies play. That's spontaneous activity. That's natural. They explore their world. They ask questions about the world. Our brain is always seeking information. It's not a passive sponge. It's an active organ that allows us to deal with an uncertain world. And if you really want to understand how we got here, how our brains got here, we really do have to start with evolution. As Theodosius Dobzhansky once said, nothing in biology makes any sense except in the light of evolution. And here is Darwin. At the end of this long spiral of billions of years, starting with bacterial single-cell life over 3 billion years ago. Now, if we look at this, the rise of mammals and then primates and humans, it's only a brief blink in the history of the universe. Even in the history of biology, what's been happening over the last few thousand years on this long line is only the last few hair width of this line. All of science is only a few hundred years old. So we're living through an explosion. What's happened in the last few hundred years is really way out of whack compared to evolution that had occurred before that. Something different is happening. Something's happened in the human brain. What is that? Now, very rarely do new things occur. That is to say, if you look at the insides of these brains, all mammals have the same architecture. Differences in sizes and the numbers of cells and so forth, but you would recognize the very same structures. If you look into the inside of a cell, almost all cells are basically the same ground plan. They use the same DNA. Different combinations of genes are being turned on, but nature is very conservative, tends to use the same genes over and over again. And similarly, as we'll see, nature also uses the same algorithms, the same computational solutions over and over again. If it works in one uncertain environment, maybe it'll work in another one. Now, for the particular question that I wanted to ask here today is, how do we make decisions in an uncertain world where there are probabilities that we have to balance? How do we make those decisions? The beginning of the story is here with the honeybee. The bee is, amongst the insects, is a champion learner. When the bee goes out to forage for nectar, it can fly kilometers away to locate a nice, juicy patch of flowers, and it recognizes and remembers the particular flower by its color, yellow here, by the shape of its petals, and the odor. And it's able to associate that by classical conditioning, by comparing the reward, the nectar, with those sensory qualities, so that the next time the bee goes to the flower patch, it can zero in on the good flowers. Now, if you're a bee, if you come back to the same patch the next day, there's no guarantee that it's going to have nectar because flowers come and go during the spring. And so you have to be prepared to change your strategy very rapidly. And bees can do that within one trial. Very fast learning, faster than humans in many cases. Now, the person who really put this on the map, bee learning, was Carl von Frisch. He did a controlled experiment in which he first, on day one, fed bees some sugar water that was surrounded by a blue plaque. And then the next day, the bees, when they came back, were given a choice. They could land at any one of these different dishes. None of them had any sugar in it. There was no differences in odor. The only difference was in the color. The rest of these were all different shades of gray. But the majority of the bees went directly back to this particular location. They remembered that they had been rewarded by a flower that previously had been, in this case, an artificial flower that had this color. Now, about 10 years ago, I was visiting Randolph Menzel's lab in Berlin. And Randolph studies bee learning by recording from single neurons in different parts of the bee brain and trying to find out what information they carry about the sensory processing, for example, responses to odor or to the sugar. Are they involved in the memory of the event after conditioning? Or are they involved in, for example, the motor side? Now, Martin Hammer, who was a postdoc at the time, had discovered this neuron. This is the cell body of the neuron, and it's called VMX1. And what's remarkable about it first is its extraordinary axonal arborization. All of this filigree here are axonal terminals, synapses, that are being formed from this one neuron onto a bunch of neurons here in the antennal lobe, which is the olfactory region of the bee brain. But that's not the only place. It also projects up to what's called the mushroom bodies, which is an important area for learning and memory in the bee. And also a side branch here to a motor structure. So here's a single neuron that seems to have its hands in 3 different important regions for learning: the sensory part, the learning and memory part, and the motor part, the action. What action should you take? And furthermore, he showed that when you give the bee sugar water to the antenna, that produces a vigorous burst of firing in that neuron. This neuron represents the reward that the bee has just gotten. Now, when I saw this, something clicked, because in my lab at that time were 2 very bright postdocs. They're still very bright. Peter Dayan and Reed Montague, both of whom have gone on to really establish this not just in the bee, but as you'll see, in other species as well. Because what they did was put together a very simple model that consisted of only a few neurons, and one in particular, this P neuron, which is the homologue of the VMX1 neuron that we just saw in function. So in the model, we get a direct input for the nectar, but we also get inputs here from the sensory inputs, for example, color, shape, odors, and so forth. And the way that this learning algorithm is organized is it's designed so that this neuron, this P neuron, which stands for prediction, this P neuron can accurately predict the reward value of a particular sensory stimulus. And the way it does it is by guessing and then being corrected. So at the very beginning, there's no knowledge. The bee has no idea what flower to land on. It lands on a random flower. Suppose it's yellow. It lands on the flower. This yellow input is active, but in fact, it's very weak so that it doesn't drive the cell by itself. But as soon as the reward comes in, the fact that it was active just before the reward means that there's a temporal contiguity there, and the fact that it was first suggests that it might causally be related to the reward. So you strengthen that synapse, and that means the next time you come to a yellow flower, and this stimulus now is able to drive the cell, this may give the bee the incentive, the motor bias, to land on that flower. And it turns out this algorithm allows you not just to mimic all of the different aspects of classical conditioning, such as secondary association and blocking, but it also allows the animal to learn a sequence of steps to get to that reward. It's not just that last step, which is the reward itself, but by virtue of the fact that this neuron comes to stand for the reward, it is an internal monitor for the animal as it goes through life in an uncertain environment of which directions to take that are likely to lead you in the direction of a reward. Now, this is a nice model for the bee, and it explains a lot of bee psychology. Leslie Reel, for example, had demonstrated that bees tend to avoid fluctuating rewards. They're conservative. They avoid risk. And it turns out that this model also avoids risk. It uses a neurotransmitter called octopamine. And octopamine, it turns out, can be used to substitute for the firing of the cell. If you pair, for example, the sensory stimulus with the application of octopamine to the mushroom bodies, this can also be used to condition the animal. So this suggests that this substance, octopamine, is a very powerful, not just modulator, but plasticizer for plasticizing and changing the strengths of the synapses according to the activity that's present in the circuit at that time. Octopamine is a biogenic amine, which is a close relative of dopamine, which is an important neuromodulator in our brains, in fact, in all vertebrate brains. There is a group of neurons here in the ventral tegmental area that projects widely and diffusely throughout the entire cortex, and very heavily, especially in the prefrontal cortex, where we know those decisions are being made. It also projects into a part of the brain called the basal ganglia, the striatum, caudate, and putamen, which are important for learning sequences of actions. And what Reed and Peter realized when they were in my lab was that it might be the case that the very same temporal difference algorithm that we apply to the bee might actually work for the human as well. Now, this algorithm makes a very important prediction that can be tested. So here's what the prediction looks like. If the stimulus comes in and it predicts a certain level of reward. Let's say it's a value of 10. And if the reward comes in at exactly 10, then you subtract the two, and it's the difference that you use for learning. If there's difference is 0, you do no learning, right? You only want to know what is the difference from the expected reward. You want to know, how good am I making the prediction? Now, if you're recording from one of these neurons, and if you're making a perfect prediction, then you shouldn't expect any difference in activity, right? It should be 0. But what if you did an experiment where you were reliably giving, say, a monkey juice, but now on one trial, where it does the right thing and it should get juice, suppose that you withhold the juice. What's going to happen? Right? According to temporal difference learning, what should happen is that you get a big prediction of this stimulus and these conditions should give you a reward. So you predict 10, 0 comes in, you subtract 10 from 0, you get minus 10, right? So with the predictions under those trials, those special trials where you withdraw the reward, you should expect a decrease in the firing of these dopamine neurons. So these experiments, in fact, were carried out by Wolfram Schultz. Wolfram is on your right. In a series of elegant experiments that he did on monkeys, recording from these VTA neurons. And before he recorded from them, they were something of a mystery. No one really understood whether they were sensory neurons or they were motor neurons or maybe something else. And he was the one who really zeroed in and figured out they had something to do with anticipated or expected reward. And here are his experimental results. So here's the very beginning of training when the reward is given and the monkey is just figuring out why it was rewarded. It takes a while for that to happen. And you can see that by looking at either the histogram, this is the number of spikes as a function of time. That's 1 second here, 1 second. You can see that right after the reward, each of these lines represents a single trial. We get a bunch of spikes about a few hundred milliseconds after the reward. And here's the peak in the histogram. That before you know anything about the contingency for getting the reward itself is a positive signal for these neurons. But after you've learned that a particular sensory stimulus, in this case a yellow light, predicts that you're going to get the reward in a short time, here a second later, now the response to the reward goes away, and now the yellow light, which before had no response whatsoever, is now driving that neuron. The yellow light comes to predict a future reward. But now becomes the critical test of temporal difference learning. Here are the trials where the reward was expected, but none occurred. The yellow light goes on, we get activity in the dopamine neuron. Here's where they should have gotten it, and there is our smoking gun. The cell stops firing for a while, suggesting that there was something here that was balanced that you didn't see. No change here didn't mean no signal. It meant that the signals were balanced. In this case here, it reveals that there's an inhibitory input that we think is a consequence of the learned association of the stimulus with the expected response. Now, what makes this really exciting and I think ultimately going to be important is that this may give us a neurophysiological handle on the question of how the brain integrates many different sensory stimuli in order to make an intelligent decision, right? Normally, if you're faced with, say, where should I go to dinner? What do? You go in your mind and you sort of think about possible places to go, and then you consult yourself and say, I think I'd like Italian tonight. Well, you may not have any choice. But what may be happening is that in thinking about these possible scenarios, you're basically comparing your dopamine response and making a decision on the basis of which you think would give your internal, based on previous experience, your internal system the greatest reward. And it's not just between different types of food. Maybe there's a choice between going to dinner or going to study, for example, if you're a student, right? Why would anybody deny themselves food to study? That seems like punishment, like sacrifice. Well, if you think that you're going to get rewarded for doing the studying by getting a good grade, and getting a good grade might mean doing well in college and then getting a good job, you may be willing to sacrifice a lot in order to get that ultimate reward. And that's something that humans do extraordinarily well. We can plan well into the future in terms of taking short-term risks and short-term losses in order for, ultimately, for our survival of ourselves and our children in the long, distant future. This dopamine system can be hijacked. All addictive drugs that are known work through elevating the level of dopamine artificially. The drive is absolutely astonishingly strong. A little bit of cocaine given to the monkeys will make them work, I've been told by my experimental friends, long into the night. They'll work for just a minute amount of cocaine, long after they've given up on juices. And rats that are artificially stimulated with an electrode that stimulates the dopamine neurons will continue to press that self-stimulation at the expense of eating or anything else until they die. That's how powerful this is. So this gives us a feeling for what may be going on under our level of consciousness. We're not aware of these representations. We only are aware of the final answer, which is, I think I'd like to go to the movie, or I think I'd like to go to the lecture. You don't really know all of the things that went into computing that answer, but it must involve, I would guess, some aspect of this machinery. Now, this insight has really transformed the way that we are beginning to think about the classic visual experiments that I told you about earlier, right? The classic experiments, you put your electrode into a part of the visual cortex. In this case, it was in the parietal cortex, and these are neurons that are driven by a sensory stimulus in the particular region of the visual field. And traditionally, the response of those neurons was taken as a representation of the physical world. That's to say, if it responded to a bar like this, that means that it tells us what's in the world that we can grab onto. But what's been discovered now is that these neurons actually are reporting on much, much more than just the physical world. And this actually is a topic that Pat Churchland, another co-author of mine, and Ramachandran wrote about in an essay about 8 years ago. And the title of the essay was A Critique of Pure Vision. And the thesis of this essay was that all the evidence is pointing toward the visual system is representing much, much more about the visual world than just vision itself. And here's the evidence. Here's the smoking gun that vindicates this position that we took. This is an experiment that was done by Platt and Glimcher. And in it, the monkey was sitting in a chair, and the monkey was getting rewarded. But the reward was changed between two blocks of trials. In the first block of trial, the monkey was rewarded with 0.2 ml of juice if it moved its, here fixated, if it moved to the right. And in the next trial, it would be getting half of that, 0.1 ml. And the stimulus is exactly the same in the two blocks of trials. The visual stimulus here produces a burst of firing, as you can see here. Here's the raster. And this is the average firing rate averaged over all the trials. For the case where the neuron, where the trials where the monkey was being given a reward of 0.1 ml. So this is a classical visually driven response, time since target onset. But now, look at the block when the stimulus is exactly the same, but now the reward is twice what it was. The firing rate of that neuron has more than doubled. This is not just a visual neuron. This is a neuron that's reporting a visual stimulus, all right, but it's also telling you something about the value of the stimulus for the animal. It's saying, on this trial, I expect to get twice the amount of reward that I did on the last trial, and that neuron now is representing that by an increase in the firing rate, and we know that the firing rate is in some way a reflection of the importance of that stimulus. So this is an interesting philosophical issue, right? What is an object? What is a visual stimulus? Is it just a pattern of light on your retina? Or does it really Is it linked up in some way to all the experiences you've had with that object? This object might be one that you've seen before, and therefore you expect to, do you know what you might want to do with it? Press the button. Or it might be a dangerous explosive, in which case you may want to run out of the auditorium, right? You would like your brain to represent those different associations because it may directly affect your behavior. And apparently in the visual system, the very same neurons that represent the shape and the color and the type of object it is also represent how important that object is to you, the value it has for you. Now, in the last brief part of the talk, I want to get back to sleep, because this is, I don't want to get back to sleep, but I think we'd all like to know why it is that we have to sleep. Every one of us sleeps a third of our lives, and that must be important. I can't believe it's just an accident. And there's a growing consensus now amongst people who study sleep that the patterns of activity that are seen in the different stages of sleep, this is EEG recordings that are taken from the scalp, which change dramatically. Here is what it looks like. It's low amplitude, high frequencies when you're awake. And as you fall asleep through stage 1, 2, 3, and finally deep slow wave sleep, it gets huge, very low frequency, high amplitude waves. And there's a growing consensus that these synchronized waves of activity, highly correlated spatially and temporally across the cortex, are very essential and important for helping you reorganize your brain, the signals in your brain, so that when you wake up, you're able to perform faster, more accurately on sensory motor integration tasks, and also be able to remember things in a more organized way. So what's the evidence for that? Let's see, I think we can come back to that in a minute. This is just to point out that during the night, this is 8 hours of sleep, you start out in the wake state, and very soon after falling asleep, you go down through these different 4 stages to deep slow wave sleep, and you stay there for about an hour. And you alternate during the night between slow wave sleep and REM sleep. Here's the rapid eye movement sleep, which when you wake someone up will report that they're dreaming something. So you can see the REM episodes at the beginning are very brief. They get longer and longer and longer as the slow wave episodes get shorter and shorter and shorter. So there's an alternation. And this must be an important clue that's telling us about what the purpose of sleep is. All of these features of sleep must have some function, but what is that? Well, here's a clue. This is an experiment that was done by Sarah Mednick in working with Ken Nakayama and Bob Stickold. And it was an experiment in which the subject, college students at Harvard, were given a task to do in the morning. And for an hour, they had to discriminate between 2 different letters, an L and a T, at the same time that they were trying to count something. So it was a distractor task. And the discrimination was a difficult one, so that they had to really do 2 tasks at the same time. Now, if they came back in the afternoon and did the task again, their performance actually went down. It decreased, it degraded. They were not doing as well as they were at the very beginning. So this is kind of strange. I mean, if there is some learning going on, you'd expect them to do better. However, there's another group of subjects that took a nap at midday. And in these subjects, especially the ones that had both REM sleep and slow wave sleep, it either canceled this interference effect, or it actually, if you had both slow wave and REM, actually improved performance. And this confirms an earlier study in which they had shown that if you come back the next day, you have an improved performance, right? So it turns out you don't actually have to sleep overnight. Just simply an hour of sleep is enough to help consolidate the learning and the memory of this particular task so that you can perform it better. So that's an interesting observation. It turns out that something similar may be happening in your brain during development, early childhood experiences. This is an experiment that was done by Mike Stryker. He was studying something called monocular deprivation. There's a critical period in the life of mammals. During the first, in the case of the mouse for this study, during the first few weeks of life, and in cats, in monkeys, and in humans, for the first few months of life, during which if you close one eye, the neurons in the visual cortex that ordinarily would have become binocular become monocular for the open eye. It's called monocular deprivation. And if this is extended beyond the critical period, that becomes permanent. Well, the question that Mike and his students posed was whether sleep had any influence on this impact, on the impact of closing the eye. So here are all of the different conditions. First of all, started out in the light, and then 6 hours of monocular deprivation. And at that point, they had different experiences. First, you can look at the animals that had the 6 hours, and you can detect already a change in the monocularity. You can also take another group and let it go on for another 6 hours, and you've discovered that at the end of 12 hours of deprivation, the change in the monocularity index is twice as big. Twice as much experience, twice as much deprivation, twice as much physical change to the brain. But now, here are the two interesting conditions. You let them sleep. You put them in the dark so that they're not getting any input. The two eyes, doesn't matter if you close the lid. One group is allowed to sleep and the other one isn't. And what he discovered was that the group that was awake but in the dark had no more change in the monocularity as the group that had only 6 hours of experience, but the one that went to sleep had the equivalent of 12 hours of experience. The experience that the mouse had just before falling asleep kept going. It's as if the animal was still experiencing monocular deprivation, and the brain just kept working along at whatever, at the same pace, imprinting that experience into the brain. And finally, this is the last example. This is taken from Larry Squire's talk that he gave yesterday at UCSD. He gave 3 lectures in 3 successive days, part of the Kuffler lecture series, summarizing a lifetime of work on memory. And this is the last slide that he showed, which is his summary of everything he's done over the last, not everything, but his worldview of the medial temporal lobe memory system, which is an important part of your memory for facts, events, autobiographical details. It's known to be important that this part of the brain, the medial temporal lobe, including the hippocampus, is intact in subjects like HM, who have had bilateral lesions to this brain area. They have very severe amnesia, anterograde amnesia. They can't remember new facts or items. They only know things about the distant past. Now, here's what Larry and others studying this medial temporal lobe now come to believe. They believe that as experiences come into the cortex and are represented here, that the changes that is labile memory, and it has to be consolidated. In the case of humans, it may take many months. And it's consolidated by virtue of the fact that the hippocampus has a reciprocal relationship to the cortex through these networks. Different parts of the cortex project into the perirhinal area that project then into the hippocampus, and feedback. And that's the feedback connections I was telling you about earlier. These feedback connections apparently are very important for figuring out how to store the information in the cortex. Now, the information isn't actually stored in the hippocampus, although you need it to consolidate the memory, because a year after you've had the experience, you can cut this part of the brain out, and you will remember. The cortex is capable itself of remembering that memory. But if you don't have the hippocampus, then you can't consolidate it in the cortex. And now, the evidence that we have, and I'm summarizing a lot of experiments done on rats and humans, leads to this picture. Here's the picture. You start out with some experience you've had during the day. And now you fall asleep and you go into what's called slow wave or non-REM sleep. Now, what's happening, we think, in the non-REM phase is that activity in the cortex, which is occurring now cut off from all sensory experience, is being driven no longer by the external world, but by the internal world, and specifically the hippocampus. Because if you record from the hippocampus during this period, it has something called sharp waves. It looks like the hippocampus is spontaneously becoming active. And this hippocampus now is driving the cortex, while the cortex is in this very slow wave sleep pattern. Now that gets reversed when you go into REM sleep. When you go into REM sleep, now the cortex, by virtue of the experience it's had from the hippocampus, now spontaneously begins to generate these memories of bizarre episodes, which are really cuts and pieces of things that have happened to you, perhaps not exactly what's happened to you, but elements of what's happened to you, but not really in any logical or rational sequence. But they're somehow related, and you can't quite make out what it means. But that information itself is driving the hippocampus, because the hippocampus goes into what's called a theta state, very low frequency, high amplitude. And that ping-pongs back and forth between the hippocampus and the cerebral cortex, back and forth during the sleep state, until finally you wake up in the morning from a REM state. Now, this so far is a very intriguing hypothesis. By no means has it been proven, but if it's true, it really means sleep is very important. One of the purposes of sleep might well be to help you organize your memory, to make things clearer, to put together ideas that perhaps are in different parts of the cortex that need to be connected. And it may account for the fact that sometimes when you wake up in the morning, a problem or a difficult puzzle that you were struggling with just before going to sleep suddenly seems to have been solved spontaneously while you were awake, while you were sleeping. And now that you're awake, it suddenly appears, the answer suddenly appears to you. Now, the very last item here has to do with what this may tell us about the awake state. And this comes from an observation by Yuri Buzsaki. So Yuri Buzsaki has recorded from the hippocampus, and he's the one who invented or discovered these sharp waves in the hippocampus, and he named them sharp waves. When the rat is locomoting, it's in this theta state I was telling you about. Again, 6 to 8 hertz, low frequency, high amplitude oscillations while it's exploring the environment. Now, the animal is awake during this whole time, but if it stops and begins to groom or feed more contemplative kinds of activities, the hippocampus switches into this sharp wave state I was telling you about. Now, these episodes can change very rapidly. 10 seconds of walking, 10 seconds of stillness, but the very same ping pong seems to be going on between the cerebral cortex and the hippocampus, even during the daylight, when the animal is actively engaged in its environment. Could it be that, in fact, this very same mechanism that we see dramatically extended in time during sleep is something that's a normal everyday experience? I mean, have you ever had the experience, you've been looking out the window, you're still, and suddenly some idea pops into your mind? Where does it come from? Well, this is the state in the rat that would correspond to the stillness. And the sharp wave, we know from the hippocampus, might well be the impulse that activated the cortex, that particular pattern of activity in the cortex that represents the idea that you just had. And of course, we always have times when we're more contemplative and times when we're more active. And it could be that sleep is just a time when you have a longer period in order to contemplate, a longer period during which you're active, but a longer period during which, for example, biochemical reactions could take place, creating long-term structural changes that then could represent memories that will last you for a lifetime. This is all very speculative, but I think it's all very exciting because we finally have the tools and techniques to actually go in and test these ideas, and the data are coming in from many different sources. So let me just end now by showing you the same picture, but now a little bit more accurately, how we think the cortex actually works. Here it is, spontaneously active. Different parts are being activated and talking to each other. That's you thinking. And now the stimulus comes in, and indeed there is a wave of activity that goes all the way up and activates all these areas. But in fact, that's not the end of it. That's not the end of the processing, because there's enough time, over 200 milliseconds, enough time for the information to go up and down a couple of times. So there's the feedback connections, and then there's the activity occurring in the earlier stages of processing. And it may well be that this feedback is very important, especially if you're trying to pay attention to something and you need to give precedence to some stimulus back here. How do you do that, except perhaps through these feedback connections? And then finally, that is processed, and you come back to your ordinary spontaneous levels of activity. It's a very different picture from the classical one, but I think we need to really come to grips with the fact that the brain is much more dynamically active and spontaneously active than we give it credit for. So finally, let me thank my collaborators, Reed and Peter. I've already told you about the work on the bee in the ventral tegmental area. I've had the good fortune to be working with extraordinarily good experimentalists, Mircea Steriad, David McCormick, who studies sleep, and Bruce McNaughton, who studies hippocampus of rats. And finally, the research that I've told you about was supported by the Howard Hughes Medical Institute and the Schwartz Foundation. Thank you very much.